My talk is called Ediths, Checkerspot, Rearing and Experiments with Host Plants. And what I want to do is I'm a PhD student. I'm in my third year right now, so I have maybe a year and some change at least left until I finish up. So a lot of my work is in progress, but I do have some kind of interesting results, and I have been rearing checker spots as some of this work. So I'll give you some details about collecting them and how I've been, what works and doesn't work rearing them in the lab. And feel free to interject with questions, although I kind of broke this up into a few different chunks. So after I've kind of talked about a subject, I'll like stop and ask if there are questions. So if you really want to ask a question, you can just raise your hand, but otherwise there'll be a little bit of time for that. So let's see, what other disclaimers do I have? So as an ecologist, I like to think of myself as kind of a generalist, which means that a lot of you know more about butterflies than I do. So if I say something wrong, you can correct me or just go like this. But my background is mostly in plant ecology and ecological restoration and management. But for my doctoral work, I've sort of been looking at the interaction between plants and insects and using checker spots as a system to do that. So, great. The outline for what I'll be talking about, I want to give you a little bit of background generally on checker spots. So I know usually Dave Nunnally gives a spiel about a given species, so today's just going to be Edith's checker spot, and I'll be your lame host for that. I don't know as much as him. And then I'll share a little bit about my experience rearing Euphydrius editha colonia, which is a subspecies that's up in the Cascades. And then we'll see how much time we have, but I want to share a little bit about a lab experiment that I did with colonia checker spots. And it has a big nasty title there, Hemiparasite-mediated interactions between plants and checker spots. And I'll explain that so it makes more sense. And then I'm also doing a field study with Taylor's checker spot. And if there's time, I'll describe that too. And these two studies I have some data from them, but they're sort of in the works. So all I can give you is preliminary conclusions, but there's a lot of cool pictures and graphs and stuff. Great. Okay, so let's start with some background. Edith's checker spot, basics. They're nymphalids, so they have 4 feet that they use to walk around with, and then 2 others that are reduced and used for tasting things, so to speak, for chemoreception. And Edith's checker spot has a huge range. It's up in British Columbia, and it's all the way down into northern Mexico. And within that species, Euphydrius editha, there are, I think, depending who's counting, something like 27 different subspecies. And they're scattered all along the west coast. So in Washington, Taylori is the subspecies that occurs in the lowlands and gets a lot of press because it has recently been listed as endangered. Euphydrius editha colonia is a more common and closely related subspecies that occurs up in the Cascades. And I'm not sure if there are other subspecies in Washington, but those are the 2 that I'm most familiar with. And they're really interesting because not only are the subspecies really patchily distributed, but within each subspecies there are disparate populations that are quite patchy. They're patchy, and they tend to be very idiosyncratic. So one population may have different host plant affinities than other populations, and it may have a different phenology in terms of when it emerges and when its flight period is. So even though they're very, very heavily studied, they're probably the most heavily studied, non-economically important insect species in the world, we still don't know that much about any given population because they're so darn variable. So it makes it an interesting thing to study. Yeah. Out of curiosity, has anybody read this book, On the Wings of Checker Spots? Okay. If you're interested in Edith's checker spot or in the Glanville fritillary, Paul Ehrlich and Ilk Hansky, who is at the University of Helsinki, I think, they wrote a book about their combined several decades worth of studying this Edith's checker spot and then the Glanville fritillary. I can't think of its scientific name. Does anybody know off the top of their head?
Of what?
Glanville fritillary. It's a related
I was talking while you were saying what fritillary?
Glanville.
Glanville.
Yeah.
I don't.
Anyway, it's not in North America, so we don't need to worry about it. But this book has tons and tons of information on checker spot biology. So if you're interested, it's worth a read.
What's the date on the book, approximately?
2004,
Okay,
I think.
thank you.
Yeah. And it's very, very substantive. Great. Okay, so one detail that I want to mention early on is the checkerspot diet. Checkerspots lay their eggs on plants that are in the Orobanchaceae family, which used to be called Scrophulariaceae, but it's the subset of that family that are parasitic plants. And then Plantaginaceae, Scrophulariaceae, and a couple of other families. And what these families have in common, they're sort of related with each other, is a group of secondary compounds called iridoid glycosides, which they're pretty common. By secondary compounds, I basically mean chemicals that plants make that are, oftentimes what it's trying to do is like poison herbivores, for example, or kill bacteria, those sorts of things. Iridoid glycosides give plants a really bitter taste and generally are thought to deter herbivores from eating those plants. However, there are a number of insects, checkerspots included, that actually specialize on plants that produce iridoid glycosides. So when they lay eggs on a plant, the female actually, we think, is tasting for iridoid glycosides. And then as the larvae feed on that plant, they sequester those chemicals in their hemolymph, in their fluids. And then over time, they will achieve a really dense concentration of iridoid glycosides in their tissues. Like there'll be about 15% iridoid glycoside, so that if a bird eats them, they taste really gross and the bird probably throws up. And then you can see here that these are not cryptic organisms. They're not trying to hide from predators. Rather, they're advertising that they're unpalatable. So like monarchs, right? You know about monarchs and milkweed. This is an analogous system. So ocuben and catalpol are two types of iridoid glycosides that are the ones that we usually talk about that get sequestered by checkerspots. But that's a really interesting part of their biology and their interactions with their host plants and the birds and parasitoids that may eat them. All right, so at least with Taylor's checkerspot in the lowland Puget Sound area, this is their food plant. One of them is Castellaea aspida, or harsh paintbrush. It grows in a number of habitats, including the prairies down by Olympia and a number of other places. Who's been to those grasslands at some point? Okay, you're familiar with the ecosystem. Good. Another thing that they eat is golden paintbrush. This is a federally threatened plant. It's not very common, so they don't have a lot of chance to interact. They used to be very sympatric, where the populations overlapped, and more recently, as both species declined, they actually didn't occupy the same sites anymore, so it wasn't an important host plant. But now that there's a lot of restoration work going on, some of the same sites now include golden paintbrush and Taylor's checkerspot, so they're starting to lay eggs on it and feed on it again. So that's kind of interesting. And then they have also expanded their diet to include Plantago lanceolata, or lance-leaf plantain, which if you gave me 30 seconds, I could run outside and get some because it grows in everybody's yard and in between the cracks in the sidewalk and all kinds of places. And it's sort of related to paintbrushes, and it has the same sorts of chemicals in its tissues, so they use it as a food plant as well, which has been, people like to argue about this because it's an invasive exotic species and they don't want it in the prairies. But it's also been useful for checkerspots because their other hosts became so rare. So it's an interesting issue. We don't really know if one host is better than the other. That's what some of my research is trying to figure out. But anyway, they feed on that as well. So I wanted to give you a little bit of a look into just the Taylor's checkerspot life cycle. So the caveat is this varies from Taylor's checkerspot populations on the South Sound prairies versus the North Sound up by the San Juan Islands and Whidbey Island. It also is different from Colonia checkerspots if you were to go up into the mountains or something like that. But the adults fly, probably the best time to see them is like the first week of May. When they do Prairie Appreciation Week at Glacial Heritage Preserve, you can get out your binoculars and see Taylor's checkerspot at that point. And the adults, they nectar on a variety of plants that are fairly common in grasslands, and they tend to cluster together. So if one emerges and it doesn't see a lot of other checkerspots, it's more likely to sort of disperse, whereas if there's a big cloud of butterflies, they'll stick together. And they lay eggs on those 3 plants I mentioned earlier: harsh paintbrush, golden paintbrush, and lead leaf plantain. And as far as we know, when they're flying around, they're looking for patches of plants to lay eggs on. And then after they alight, they use their chemosensitive forelegs to touch the plant and taste it, so to speak, to figure out if it's the right host and if it's got the right chemical makeup. So, yeah, and it's a really pretty butterfly. In terms of identification for Euphorbia editha, this line right here is referred to as the editha line, and if it's got some orange in it going all the way down the hind wing, then you've got Euphorbia editha. So that's how to tell it apart from many other Euphobias that it can co-occur. All right, so the eggs. What's really interesting about checkerspot eggs is they're laid in clusters. Most butterflies lay single eggs. Checkerspots dump a load of eggs all at once. And folk kind of debate about why, but they tend to lay a cluster. If they just started flying, they may lay 150 eggs on one plant. The normal amount is maybe 40 or so eggs. And they tend to lay them on the underside of leaves, and they take 1 to more like 2 weeks to mature, depending on the weather and things like that. And they're these little barrel-shaped tan eggs. They're bright yellow when they're first laid, and they turn tan like this. And then later they darken. And I've got some pictures of that from the lab. The eggs hatch, and then we have larvae. So their larval cycle, they feed for a few instars, and then when things dry out in the summer, they go into diapause. And they basically just go lay in the thatch underneath vegetation for like 8 months. And then they come back out and feed again. So this pre-diapause larvae period, the hotspot is kind of in June. So when things are starting to warm up. And in this stage, since we had a cluster of eggs, we have a cluster of larvae that feed on their host plants. So they spin a web around the plant and they feed in there together, and that's a way to avoid predators and create a favorable microclimate so that they don't dry out, because they're out in these open, sunny environments and it can actually get really hot. And part of what that means, when they're really small, they're incapable of leaving the plant that their egg was laid on, because their whole world is just a couple of centimeters. So in this early stage, they're basically trapped with all their siblings on one plant. So it better be a good one, and it better be big enough that they don't eat it all and starve. And here's what they look like when they get a little bigger in the 4th instar. And at this stage, they're less gregarious. They don't hang together as much. And once they're towards the end of their 2nd instar, or maybe their 3rd, they start to wander about a little bit more. They move maybe a meter and move from plant to plant and taste a plant. And that one's not good enough, they go to a different one, that sort of thing. And once they get to their 4th instar, they're able to crawl down into the thatch and go to diapause. So, and then in terms of diapause, this is a really mysterious life stage because we can't find them. We don't even know. It's really hard to find a diapausing little black caterpillar. And they spend like 8 months just sort of down in the thatch. A lot of them die during this stage, and it's really interesting to talk with people who manage grasslands about how to do controlled burns and how to they do different herbicide treatments sometimes to remove the invasive exotic plants and stuff. So, and what happens to those larvae while they're in diapause, we don't really know. So that's an interesting area that needs more research. After diapause, sometime they spend 8 months just laying there, right? That's what they do for most of their life, is just sort of build a little web and lay down on the ground. When they come out, it's sort of during the winter green up. So usually like late January and February and March, they're out as these much larger post-diapause larvae. And at this time, they're able to walk all over the place. They can walk like 10 meters, and they do a lot of thermoregulating because, as you know, in April, occasionally we have a beautiful 60 or 70 degree sunny day that if you're a caterpillar, you can go eat a lot. And a lot of other days, not so much, right? If it's below about 50 degrees, a caterpillar really can't go out and eat anything because it's cold-blooded. It's an ectotherm. So they're very dark, and they spend a lot of time positioning themselves on the south sides of slopes, trying to get enough sunlight that they can go eat something. So they do that in the spring for a few months. They also nibble on a couple other species of plants that are related to their other hosts that are annuals, so they're only available early in the spring. And then eventually they wander off and they pupate down near the soil surface on the undersides of rocks and dead leaves and things. And then the cycle continues. So here's what the chrysalis looks like. I think it's a really beautiful chrysalis. It's sort of cryptic colored. But I love how you can see the wing patterns in the abdomen sort of on the outside.
And what
Yeah.
size is it?
I'd say it's about 1 inch long. It's about as big as this last knuckle on my pinky.
Thank you.
And the final instar larvae are maybe a little more than 1 inch long. So, yeah.
You mentioned the post-diapause annuals that they use, like Colensia and Plectritis.
Yes.
Are you aware of any preference for the other host plants over any preference for one group or another, or seemingly
Yeah.
random?
What I've noticed is in the little pilot experiments that I've done with post-diapause larvae, they spent a lot of time eating either plantain or paintbrush, and as they walked from plant to plant or patch to patch, as they sort of exhausted it, because they start to eat a lot, so they will eat a whole plant. On the way, they would stop and nibble on these other species.
So it is indeed nibbling. I mean, that is,
I would
it
call
is
it
indeed lesser eating of those annuals, Plectritis and Colensia, than it is feeding on the Plantago and the Castellet.
Correct.
Thank you.
Yeah, they're like appetizers and desserts, not the entree. Great. Okay, so that's just my little intro into a checker spot life cycle. So does anybody else have questions at this point?
So did you suggest that during that long diapause they're developing or growing in some way? That
They're not.
Smaller when they go in and they can walk more when they travel farther when
They sometimes they'll advance 1 instar while they're in diapause, but they're quite a bit smaller when they come out of diapause because they lose so much water over the process. And then I think once they sort of warm up in the spring and they start feeding, and really quickly their mobility increases. They don't like wake up from diapause able to run 10 times faster or anything like that. Yeah, good question. Any other questions at this point? Oh.
Yeah, during that diapause, it seems that if you're doing controlled burning, they'd be extremely vulnerable.
Oh yeah.
So do they time the burning earlier or what?
What they try to do is they do surveys on a lot of these populations because there are so few of them, so they know that the bulk of the larvae are, say, on the west side of the preserve, so they'll burn the east half.
Okay.
And then eventually, if they find that they're mostly on the east side, then they'll burn the west. What actually happens at some of the prairies that are on Joint Base Lewis McChord, they're in the artillery impact areas. So that means there are like Army people doing drills and blowing stuff up in the checker spot habitat, which sounds horrible and might kill some, but what it also does is start a fire every year, or multiple fires every year, which means every time there's a fire, it's actually, it just sort of smolders and burns a little bit at any given time, meaning there's a ton of, it might knock out a few, but it mostly doesn't harm the butterflies. So if you had a really, really high frequency, low intensity fire, it probably wouldn't be as big a deal. But the way that we have to manage most prairies, which is like this, this big wall to wall, violent fire thing, that's got to be very, very harmful if it were to hit diapausing. Okay. Great. Okay, moving on. I'll share now my experience with rearing Euphydrius editha colonia. So this is no longer Taylor's checker spot, but it's its closest relative subspecies, which occurs up in the Cascades. Two resources that were super helpful to me. One was the Life Histories of Cascadia Butterflies, or I always accidentally call it Life Histories of Cascadia Checker Spots, which would be a much more specific book, but by Dave and Dave. That's a really helpful book. And then, since Taylor's checker spot is endangered, they actually have a captive rearing and release program, and it's based in the Oregon Zoo in Portland and at the Mission Creek Women's Correctional Facility, which is out on the Olympic Peninsula, which is part of a program that they do with Evergreen College. And they capture butterflies and then basically propagate them in an indoor facility, much like what I did for my research, and then they release post-diapause larvae out into prairies. It's really cool facilities. I've been to both of them. That's really interesting. It's also cool to see inmates super motivated about butterfly conservation and doing really good work producing lots and lots of caterpillars to put out and try to recover some of the Taylor's checkerspot populations. But anyway, they also produced this manual, which is really useful if you want to know how to rear checkerspots. Okay, so I wanted to have a population in the lab for a little while, a colonia checkerspots, so that I could do some experiments in the greenhouse that I'll get to later. But so basically I just wanted to go collect some butterflies and have them lay eggs and rear their caterpillars. So I went out to Quartz Mountain at the recommendation of Dave Nunnally. He gave me directions to this beautiful, beautiful spot. So this is sort of near Ellensburg. Has anybody been there? It's gorgeous, isn't it?
I think we've all been there.
Oh, you've all been
It's
there? Oh, okay.
a
Super.
field trip every summer.
Okay, it's a great spot. So I went out there and collected a bunch of checkerspots and sort of weeded through them and found 10 healthy looking females and then brought them back to Seattle. And what I found worked really well for feeding them was a mixture of water and buckwheat honey. And I'm not sure why buckwheat honey. I think it has a lot of minerals in it. But
What else did you try?
oh, I just read that that worked well, and I tried it, and it worked, so I stuck with it. And it's really interesting. The first couple of times you try to put a Q-tip in front of the butterfly, they're like, well, that's not a flower. But after a while, it's almost like they sort of trained to it, so that I could hold it next to them, and their proboscis would dive right into that Q-tip. So it was kind of neat to see. And my mission with these 10 female butterflies was to get them to lay as many eggs as possible. So what I would do every day is feed them some buckwheat honey, and then they all had their little egg-laying chamber. So this is a pot that had some Plantago or Castanea in it. I wish I could show you. I did this last summer in 2014, but this all happened right in the buildings next door here. I would feed these butterflies and then put them in their little enclosed pot with a plant that they could lay eggs on. And then I put them in these cool zippered enclosures, and I brought some because I think they're really fun. And they're sort of spring-loaded, so they. You guys, do you use these things? I love these. They're super handy. So if they cost like $20, you can get them on the internet.
What are they called?
What is the name? Rearing cages.
Just very
Yeah, I think this one's called a butterfly farm. And then they have a bigger one. So this is the one that's in that picture there. And I had one of these sitting in my office today because I wanted to bring one just to show you in case you hadn't seen it. And occasionally when they're all coiled up, they just go poof. So I was sitting working on my computer and it went wow. And expanded itself. But these are really handy tools. So if you ever want to borrow one, I have like 100. Great. So what I would do, if you can see in here, here are all those little pots with a butterfly in them, and I wheel them out into the sun. And I had a nice streak of weather where it was sunny and maybe 75 or 80 degrees. And for these female butterflies, if you give them nectar and then warm, dry weather with a plant to lay eggs on, they will lay eggs. So a lot of them started laying eggs every day for maybe a week or two, which was great. So I would just wheel them in and out of the sun every day.
Wait, you skipped the whole mating process.
They were already mated when I, if you find a female out in the field, odds are it's already mated.
Okay.
So of my 10, 9 of them laid eggs.
Okay.
Yeah, I was lucky I could just get that. Yeah, but I think the males come out first and sort of patrol, so as soon as the females emerge, you can figure they're pretty much mated. Yeah. I was glad I didn't have to. I tried that later on the next year, just kind of for fun, but yeah, it's a little trickier to get them to do that. Butterfly matchmaker. Yeah. So here is a photo of one of the females ovipositing on golden paintbrush.
Usually on the underside of the leaf?
Yes. Usually, not always, but usually towards the bottom of the plant on the underside of the leaf. So this is very typical. They would hang on the leaf and then curl their abdomen under and then lay the eggs in rows. And then if they sort of built 4 or 5 rows, they'd start stacking eggs on top of them, which made it really hard to count, by the way. So here are what some of the egg clusters look like.
Oh.
Yeah. So they're beautiful bright yellow at first. And this was my setup. I can go into more detail if you all are interested, but basically I just had a wire cage. I got some shop lights at the hardware store and put full spectrum bulbs in it, put them on a timer so that the sun cycle was similar to how it was outdoors at that time. And our lab's about 22 degrees Celsius, which is great. And then I put the eggs in, I brought some bins just to show you. Yeah, these shoebox bins are great. And then these are great for eggs and early instar larvae. And I just got them at Fred Meyer and used a needle and poked a bunch of holes in them. So pretty basic stuff. When I had a whole lot of larvae, I used these Ninja Turtles ones that are a little bigger. They were on sale, so they have Ninja Turtles on them. They were cheaper. And then I put those in these shoebox bins and just put some paper towel on the bottom and squirted water on them every day to get a little more humidity because we don't want things to dry out. And that worked pretty well for me. I went to Joann Fabric and I got some, I don't know if this is for making tutus or what. They gave me some weird looks because I was buying armfuls of this stuff. But put a rubber band over there and you're set to go. Yeah, so that was what my setup looked like. And I borrowed this photo because I forgot to take pictures of this part from the manual. But if you sort of make a little funnel of paper towel, then when the eggs hatch, they've got food there to eat, and when they defecate, all the frass collects at the bottom, which is handy. So here's what the eggs look like right before they hatch. And I've got a lot of pictures with the black, like a circle like that, and that's from me taking my camera and pushing it into the eyepiece of the microscope in our lab. So I've got a lot of those. And I've got some cool videos I'll show you too that look very similar to that. But the eggs take this sort of eggplant-looking color right before they hatch. And then here's what they look like when they hatch. Those are the first instar larvae they eat their egg casings first before they move on to the plant.
And they're very tiny.
They're very, very tiny. They're
Fingernail. Like the fingernail.
less than a ½ centimeter. Yeah, end of the fingernail. They're very, very small. So yeah, then they do pretty well living in these little tubs. I found that feeding them plantain was much easier than castellea because plantain's really weedy, but seeds don't have any dormancy. You could just take some and throw them on some soil in the greenhouse, and a week later you've got a forest of plantain to pick. So I mostly feed them plantain if I can. And basically all I did was swap out the food every day, take out the old leaves and put in new. Every couple of days you need to clean the frass out of the containers. And as if you had a cluster of like 150 eggs, once you have 150 larvae, at some point you need to split them into smaller groups. So these are still in their first instar here, very, very small larvae. I mean, you can see that's an egg, which is about the size of like a typed period at the end of the sentence. So
that's magnified.
very magnified through the microscope, yeah. And I found paintbrushes are a great way to move them around. So this is a second instar larva on a very fine little watercolor paintbrush there.
Do you know what kind of brush hairs that those are?
No, I think I was supposed to use something natural, and I just
But
used
it
what I
worked.
had.
You used what you that and it worked.
Yeah, it worked fine. I think as long as they're not too stiff, because you don't want to like puncture a caterpillar. That would be unfortunate. So here's what they look like once they're, this is kind of a big cluster that's been skeletonizing plantain leaves. And if you catch them on a hungry day, they can eat a lot, which is really interesting.
And that's all their waste? All their
Yes, that's
waste?
all those little black dots are frass. Yeah, so it's a pretty poopy ordeal, really. And you gotta clean it out because it'll get moldy and it gets gross. And here's what they look like in, I think this is right before they get to their third instar. So at this point, they're probably a centimeter or a little smaller in length, so quite small. And here's one that's just about to molt from second to third instar. And here's its old exoskeleton there. And here's one that just came to third instar. They have this really funny sort of tan coloring when they first emerge, which I'm not doing anything academically looking at how they molt or the physiology there. I just thought it was really cool. So actually, I'll show you some videos of them molting that I have. Fun to watch. And I caught one like a mug shot. So I thought that was kind of cool. So they're just so fascinating when you look at them up close. Okay, so what I noticed with their they would advance from one instar to the next in, depending on the instar, maybe 5 days or so. And what that would entail, they would eat really quickly for maybe 3 days or so, and then they would slow down, and you could tell something's going on inside of them, and they're not hungry because they're ready to shed their skin, move on to the next stage. So they would slow down for a couple of days and become inactive, and then I would know they were about to advance. So I want to show you a video that just has some of the early stages, and I just sort of edited it together. It's videos that I took through the microscope, so sometimes it gets a little jostled, but you can see them feeding and moving from instar to molting. So it's kind of cool to watch. And for some of them, I sped it up a little bit. I figured out how the video editor on my computer works, which was fun, and increased the speed so that they sort of burst out of their skin, which I thought was fun. So I'll narrate this a little bit. And there's no sound. So these are just first instar larvae. So this is very magnified. Just walking around.
Do they ever eat each other?
I never saw them eat each other. Good question.
It's like you checked somebody out, and
Yeah.
then just went somewhere else.
And these are still in first instar eating plantain. So you can get a sense of, those are the hairs on the plantain leaf. And they're very large here. And you can see right through, they're translucent, so you can see their gut is full of green stuff.
Oh, that's their inside.
I believe so.
Wow, that's amazing.
Kissing.
Yeah.
So what purpose do the hairs on their own body serve?
I think it's a protection for parasitoids. So this one is moving from first to second instar, I think. So it's crawling out of its skin. It's trying to pull its old head capsule off.
Oh, wow. Oh, that's.
So is this speed enough?
This is sped up about 4 times. So you can see they're all sort of jittery. It's sped up. And then another one's going to go too. Yeah, they have to pull their helmet off.
They all mold in a time, narrow band of time together.
Often they do. It's fairly synchronized.
Yeah.
So these are from second to third instar, I think, now. So you can see they're getting bigger and darker, and they've got those characteristic orange spots now.
And spines are getting complex.
Yeah, it's really actually, this one, I think, they sort of flex their body in order to make their spines, which are folded up, sort of top out. So I think this one you can see it pretty well. It'll sort of stop and do these little stretches, and you'll see its spines expand. There.
Oh, wow.
Got me an itch.
Yeah, it makes you itchy.
It looks like he was moving the spines intentionally.
Yeah, I don't really know how it works. This is the last one. I think this is from second to third instar as well. I'm not sure.
Silvery.
Yeah, it's like clear.
Wow.
That's so
And does their head capsule, it looks like it's black every time, so they change
Yeah.
after he
It
gets rid of the black
darkens.
one? It darkens.
It darkens over time. So you can tell when they've just molted sometimes because they have this tan look to them, and then they harden up and turn dark. Yeah. Cool. So I thought those were really fun.
Great.
And I wish I had more. I tried to take some time-lapse video of like eclosion from crystals and stuff, and it didn't work very well, unfortunately.
Do they
So-
eat their skin at all?
I never saw them eating their skin, although I wouldn't either. I mean, they eat their eggs after they hatch from them, so I don't know. Yeah. Okay. So at some point, once the larvae get to about their fourth instri, they stop feeding and they get sluggish. And then eventually they sort of climb down to somewhere sheltered, which in a paper towel environment tends to be the bottom of the paper towel. And they spin a little web, and then they just sort of lay there. And then they do that for like 8 months. So what I did for fall, while the temperature outside was still really variable, I just left them in the lab. And then I moved them outdoors to, this isn't our facility because I forgot to take a picture of it, but it looked exactly like this. Just take some pallets and some terracotta pots and flip them over, and then put the diapausing larvae in their tubs inside of those pots, and that creates a nice cold but not too crazy variable environment for them to be in. And it's in a sheltered area so they don't get rained on directly. They're just sort of bathed in the winter mist.
Yeah, it's dark.
Yeah, it's nice and dark. Yeah. What I found for larvae that I reared on plants in greenhouse pots is they would always diapause in the lip of the pot. So they would walk around all over the place, and they'd climb in the little fold on the edge of the pot and spin a web and close it off, and just sort of sit there like this. You know, sometimes in groups. So I thought that was kind of cool. Since I was dealing with caterpillars that came from a subalpine population, spring in Seattle comes before spring on Quartz Mountain, obviously. So what I did is I moved them indoors to a winter growth chamber that's just like a little refrigerator box that's set to refrigerator temperature, basically. You can just put them in a refrigerator like that. But I used a growth chamber because I'm a scientist. And then in June, we moved them back into the lab and gradually increased the temperature and gave them food plants. And quite a few of them died during diapause, but some of them.
What percent?
For me, it was probably more than half of them, which is a lot. It used to be a really high percent in the captive rearing for Taylor's checker spot, but they've, by sort of tweaking year after year, now they're down to less than 10%, I think, dying. So, it was my first time.
So, but, I'm curious about that because if they were outside, there's always the possibility of predators.
Oh, yeah.
And you didn't have that issue, but I'm sure that there's variabilities in the temperatures and outside and, they can freeze to death or...
I think most of them die during diapause. I mean, that's, one butterfly lays 1,200 eggs, and
Okay.
most of them die at some point. So I think a lot of them die during diapause, honestly. Although, there's not a lot of data about that because it's really hard to study. Once they emerge from diapause, I just put them in these little tubs here and put some leaves on one side and a little ball of paper towel on the other so that they could run around in here and thermoregulate and climb on top of the mound when they wanted to sun themselves and climb down and eat. And that seemed to work pretty well. And here's what they look like after diapause. So at this point, they're a couple centimeters long, a little more than an inch. They move around a lot. We had, I think, at least one that got out of the bin, and I found it like 30 feet away on the other side of the lab, looking for food. So they're pretty adventurous at this point. And then they would crawl up on the mesh and pupate. So here's what the pupae looked like. I had one that fell off, so I got it under the microscope. I just think they look really cool. You can stare at them for ages. And then after about a week, they would declose. So here's one having its first meal in our lab after coming out. They're gorgeous butterflies.
They're so perfect when they first
Yeah, sort of spotless. And I experimented a little bit with mating them, and I had some success with them, but I didn't end up maintaining the population. My experiment was over. But if you put them together in an enclosed area, they'll figure it out. That's what I found. It's biology.
So did you take them back to Quartz Mountain or?
No, we did some chemical stuff trying to measure the levels of iridoid glycosides in their bodies. So at some point, anything that was surviving was ground up for lab tests. So sad. Great. So that's sort of my experience with their life cycle. Any questions about that?
What happened when you studied the chemicals in them? What did you find?
A lot of that's still being analyzed, but one question, we didn't even know if Colonia checkerspot or Taylor's checkerspot actually sequester iridoid glycosides, or if that's something we know about checkerspots in general, but we can't actually say that that occurs in every population. So now we've done at least a little bit of analysis with them, and we know that their bodies can contain up to like 15% iridoid glycosides, which is pretty high. One of my committee members, who was the scientist who figured out that that's what they do and that's how they interact with their host plants, she said I think in the 70s, she ate one of the caterpillars to see, and she agreed that it was indeed very bitter.
Did you use other kind of butterflies or other species to know if it was more bitter than?
Yeah, she had, I forget which common species she used. I think it was painted lady or something like that, or no, woodland. And that one was tasteless, and the checker spot, I think she mostly was working with Baltimore checker spots, which are the common checker spot on the East Coast, and found that they tasted awful.
A question on the glycosides. Have you found out yet if they vary depending on the host plants, or is that coming up?
A lot of that is still in progress, so I can't say anything definitively. I can say that, so we'll get to the idea that paintbrushes are actually parasites on other plants, so that other plant can influence the amount of iridoid glycosides that paintbrushes produce. And that we have measured. And yeah, I think generally the 3 host species that Taylor's checkerspot uses, they all produce roughly similar iridoid glycosides. But we have a big experiment that's still running that will tell us more in a year or 2. So unfortunately, I can't give too many details yet. You'll pick up some tidbits along the way here. Great. Okay, well, the next little stage here, I want to share a little bit about the experiment that I was doing, that I used all these larvae for. So what I was studying was the ways that checkerspots interact with their host plants, particularly Castanea, and the ways that they indirectly interact with other species. So this isn't really a conservation question as much as a theoretical ecology question. So it's not all that useful, but I think it's really cool. So indirect species interactions, is this a familiar term to many of you? Okay. How about trophic cascades? Okay. Well, we all know that species interact with each other directly, but what we are less likely to think about is that if you have one pair of species that interact with each other, and then one of those, if species A and B interact, and species B and C interact, even if species A and C never cross paths, they can influence each other by changing species B. So an example, common examples are, like in Yellowstone National Park, the wolves were removed, and the wolves eat the elk, eat the plants. So when there weren't any wolves, the elk got overpopulated, and they all went out rampant and ate a lot of vegetation, and they ended up causing stream bank erosion on the edges of the Yellowstone River and all kinds of things. Eventually, people, this was very contentious, but put the wolves back in Yellowstone, and the wolves changed the number of elk and changed the behavior of the elk, and the elk stayed away from the stream banks, and the aspen regenerated, and it changed the Yellowstone River probably back to a little bit more like it used to be. That's a really commonly cited example of an indirect interaction. Another one that's related especially to University of Washington, a lot of ideas about keystone species were developed by Bob Payne and others on the Pacific coast. So for example, sea otters eat sea urchins. They love eating urchins. And urchins like to eat kelp. So if you've got sea otters, everything's in check. If you remove the sea otters, the urchins go nuts and they eat all the kelp forest, and then you end up with a barren instead of a kelp forest. Put the sea otters back, you get the kelp forest back, even though the sea otters are not planting kelp, right? That's the idea here. And I was interested in looking at ways that plants interact with parasitic plants and the animals that eat parasitic plants. So, if this is a paintbrush plant, excuse my hand drawing, below ground, they connect to other plants in the community with these little things called haustoria. And they're basically like little vampire teeth suction cup things. and they sucker onto the roots of the plants around them and then pull stuff out of them. So they're called hemiparasites because they're green and they do some photosynthesis. So they're able to survive on their own, but they're also stealing stuff from the plants around them. And what that looks like through a microscope is sort of like that. So you see the root and then it has this little suction cup thing and it grafts itself into its neighbor's root systems and it steals water and photosynthate like sugars and nitrogen and even secondary chemicals and stuff. So super interesting.
Is the plant on the left supposed to be something specific?
Oh, I was trying to draw Romer's fescue, but they are generalists, so they will parasitize not everything, but most of the things that they come across. And that can result in some cool indirect interactions. So for example, you can have a paintbrush plant that parasitizes a host plant, and by maybe changing the number of flowers that the host plant can produce because its resources are being taken away from it, it can reduce the seed set because less pollinators visit it. It can change the relationship with herbivores because the plant is weakened and then more herbivores are able to come and eat that plant. Parasitic plants can also reduce the abundance or cover of a plant that they're parasitizing, and then if that plant was really competitive, it's less competitive, so other stuff can move in. So even though the parasitic plants might not be interacting with that other stuff, they're sort of facilitating it by suppressing their host. And then the interaction I'm interested in is whether a host plant can change a parasitic plant and then change its relationship with the herbivore that's eating, which is Checkerspots.
And are parasitic plants really common?
They're in most ecosystems, so paintbrushes and what else, pedicularis is parasitic. It's like 1% of plants are parasitic, something like that. So they're not everywhere, but they're in most ecosystems, and sometimes they can be really important. In European grasslands, for example, there's a parasitic plant, rhinanthus, that whether it's present or absent, it'll totally change the plant community because of the plants that it's sort of suppressing, and it changes the way nutrients cycle because it's stealing nutrients from plants and then decomposing on the soil surface and stuff. Yeah. So they're really interesting and kind of mysterious. So my little hypothesis for this project was, hey, if we change the identity of a single host that the paintbrush is parasitizing, so I tried yarrow and Oregon sunshine and Romer's fescue and I can't remember the common name of this champs yet, and plantain and a lupin, and I grew paintbrush that could parasitize each of those. And if we did that, we would expect the paintbrush's traits to be different depending on which host it has. So maybe it grows bigger with some hosts, or its leaves have more nitrogen, or it produces more or less iridoid glycosides, depending on which host it's interacting with. And then, if it's interacting with a caterpillar, maybe we would see differences in how many caterpillars survive, or how big they grow, or how many, whether they're able to sequester iridoid glycosides, all those kinds of things. Even though they're not interacting directly with the Castellia's host, they can be indirectly affected by it. So that's the idea. And I made a little experiment in the greenhouse where I grew golden paintbrush. I just picked golden paintbrush because my lab does a lot of stuff with golden paintbrush, so that's one I used. And grew it with each of those 6 species. So this is Oregon sunshine here, Eriophyllum, and this is Romer's fescue. And I grew those plants up. And then on each of those pots, I put 5 second instar Colonian checker spot larvae.
The one that you had raised.
Mm-hmm.
So you raised them for this?
Yes.
Oh.
Yeah. And some other stuff, but for our purposes for this. Yeah, and I couldn't use first instar larvae because they're so darn tiny, and because they desiccate if you pull them out of their web and go stick them in an experiment. So I had to wait until they were second instar. So here's all these pots with Castellia growing with different things. Here's Castellia with lupin on the right. And then I put each pot in one of these cages. So there's Castellia and yarrow. And then I had a whole lot of those cages. So every one of those cages has a pot with a paintbrush and host plant and 5 larvae eating the paintbrush.
And how many are there?
I think there were 72, something like that. Yeah. So it was a big mess in the greenhouse. This was summer of 2014. And here's larvae that were just introduced to a plant, tasting the tip of the leaf there.
That's a beautiful picture.
Thank you. And so I basically left them to their own devices in the pots, water the plants and let them grow. So here they're really probably fourth instar there on the right. See, they make a total mess of the plant and grass everywhere. And then I took a bunch of measurements. So I was interested for the paintbrush in how long the stems got and how much the plant weighed at the end of the experiment. And then I used some what's called a CHN analyzer to figure out what's the concentration of nitrogen in all the leaves. So if it's different depending on which host. And then the leaf iridoid glycosides, which Dean Bowers is on my committee. She's at University of Colorado and is the global iridoid glycoside leader person. She knows all about that. And then for the caterpillars, I just kept track of how many survived, what they weighed at the end of the experiment, and then their iridoid glycoside concentrations as well. So first, let's look at what happened to the paintbrush. You can see just a visual example. On the left, it's growing Chromis fescue. That plant doesn't happen to be very big. It's not flowering. On the right, you've got a big, hulking, like meter-long paintbrush that's growing with yarrow. They just love growing with yarrow for some reason. And here's the total stem length. So the take-home message is, here with yarrow, they are growing more than a meter long on average if you add together the length of all the stems. That's a big castellea versus really small on some of these others. Pretty big on organ sunshine. And then here's the mass of the castellea, which is pretty much the same. I could toggle back and forth. Pretty much the same. And then the nitrogen content. So this is sort of like, is the leaf more like spinach or cardboard? Right? More nitrogen, more like spinach. You can see again with yarrow, really nitrogen. Yeah, question.
So I see Castellea on the x-axis there. So
Oh,
you're
yeah.
saying they can parasitize each other?
No. So good question. Thank you for asking that. I also included a control where I had castellea growing with no host. So what I did is I paired it with another castellea so that there was still another plant taking up space in the pot. So that's what that is. Yeah, thank you. But I've never actually seen them parasitize each other. Great. So you can see there's a variation in nitrogen content as well. So they're nutritionally different. And I still don't have the lab analyses for the iridoid glycosides in the plants. So that's thank you. Yeah, castellea are great for that. So here's what happened to the larvae. Here's their survival. If they were on a paintbrush that was paired with yarrow, like 2/3 of them survived. If they were paired indirectly with Deschampsia, for example, like 6% survived. So huge difference in survival based on which plant paintbrush was paired with, which is kind of interesting.
So it looks like on with the Deschampsia that the performance is less than with the other than with the control. So why do you think the parents? Why do they do?
Now that's a good question.
So yeah.
This isn't
Like you say,
statistically. These two are not significantly different from each other, even though their means are a little bit different. So there's that. I think it could also be like if you put two castellea in a greenhouse pot and they are competing with each other a little bit and they're both pretty small, versus if you put castellea in a pot with Deschampsia that sucks up all the water a lot more than a castellea would, and it's not a very good host, so then the castellea actually does worse. But yeah, it's a funny result. We get that a lot, where castellea actually does worse with some hosts than it does by itself. Yeah, go figure. Another question?
And then so similar questions before. I see plantain on that list. Is that plantain by itself or is that plantain as a host?
Thank you for asking. That is plantain as a host, and since the caterpillars would just as easily crawl off the paintbrush and go eat the plantain, I used some more of this mesh so that the plantain were pinned down under the mesh and just the castellea was poking out, so they could only eat the castellea. I think there was one pot where they had pulled through the mesh.
Kind of a torture on them.
Yeah, there was way better food right under than that. And here's the mass of the caterpillars. So it's a little bit different now, but they got a lot heavier when they were with yarrow and with the castellea that were on Plantago than with the others. So there's some, it affects their survival and it affects how big they can grow. Not really in very predictable ways.
Extremely variable.
Yeah, really variable. And then here's the iridoid glycoside concentrations in the caterpillars. So this is the sum of all the different kinds of iridoid glycoside. And what's really interesting is with some of these, a lot of them die and they don't grow very big. but they're chock full of poison, which is great for them if there's birds around, right?
If they haven't tried first.
Yeah, right. So if they make it through the gauntlet, they're chemically defended really well. Whereas if they're eating this beautiful paintbrush plant that there's so much of and they grow really big, they're actually not able to sequester a lot of iridoid glycosides. So they're this big, luxurious caterpillar, and like you could eat it and it wouldn't even taste bad. So that's really interesting.
But
And
on the Plantago, they were growing bigger and getting.
They survived okay, and they grew pretty big, and they got a lot of chemical defense. Yeah, so really interesting.
I was just wondering about that broad uncertainty bands. Is that a function of the experimental measurement or large variation in samples?
Yeah, so this is the mean, and this is one standard. Is it standard error or deviation? That's
Yeah, so
bigger. It's the bigger one.
Yeah.
So yeah, there's quite a bit of variation, which is these letters here indicate whether they're statistically different. So if they don't share a letter, then they're different from each other. So these two, since they both have a C, are not different. This one, it differs statistically from this one and this one, but not this one or that one. Yeah, they were just really variable. So there was a lot of discrepancy in terms of the size of the larvae in some of these and the survival and also the chemical concentrations. So there was also a lot of discrepancy in terms of how big the paintbrush grew. So I guess it's just
variation in a lot of it had to do with variation in the paintbrush itself.
I believe so, yeah.
Yeah.
Yeah, so the results aren't super neat and clean, but they're also pretty significant. So it's been interesting. I should say this is preliminary. I just got these data. I haven't really sifted through them that much. What I thought was really interesting is there's a bunch of studies, a couple of studies at least, that show that when plants that produce iridoid glycosides, if they're fertilized or if they grow in a really fertile environment, they don't produce as many as their concentrations of iridoid glycosides are much lower. So like, for example, I tested some paintbrush that were in plots that hadn't been burned in a couple of years and plots that had just been burned. And the plots that had just been burned had less than 1% iridoid glycoside concentrations. The ones that had not been burned had like 5 to 10% iridoid glycoside concentrations. So it's a huge difference depending on the environment the plant's growing in. And similarly, there's a huge difference in Castanea depending which host it happens to be parasitizing. So I thought that was really interesting. And I still have to do a lot of statistical analysis to link together. This is, for example, the leaf nitrogen concentration of the plants, and then how big the larvae are. You can see there's probably a positive relationship there, but it's really messy, and I'm going to have to do some really complicated statistical stuff that I'm dreading. Yeah, structural equation models. Yikes. So some basic conclusions we can draw right now. There's definitely a strong indirect effect where hosts change paintbrush and it affects the larvae, which I think is really interesting. It affects their survival and how big they grow and their chemistry. So that's kind of interesting. And generally, parasitic plants do really weird things in food chains and food webs, which I think is really cool. One question, this was sort of a preliminary study, a pilot study. I don't know what importance this has out in the field, because you never know what the Castanea happens to be parasitizing, and they parasitize multiple things at the same time. So it was a good introductory study to say, hey, wait a minute, does Castanea's host affect the caterpillars? And the answer is, yeah, but we don't know exactly how that would bear out if you were to go out into a grassland, for example. So it's the first step.
So could you do experiments where it's parasitizing several plants and see what happens then? That would be more complicated, but it could be done.
Yeah. We actually just submitted a grant to do that and to change the diversity level of the host community. So what happens if you have a bunch of Castanea and 16 plants that are all the same species, or 2 species, or 4 species, or 16 species? So we would love to do that in our lab, and we asked the government for money. I don't think we're going to get it. But, So we're waiting. We'll hear around Christmas. But if that happens, I'm going to stay for like 3 more years and do that experiment. We'll see. Intermission. Okay, so the last thing that I'll talk about, and I'll run out of time here, so I'll breeze through some stuff, but I'm also, this is actually the biggest project that I'm working on, and it's maybe about halfway done right now with the field work. So again, I can just sort of share some tidbits and not really the whole take-home message yet, but it's kind of a cool study. So what we're interested in is figuring out what's the relative suitability of the different host plants that Taylor's checker spot uses. So there's 3 hosts: plantain, and harsh paintbrush, and golden paintbrush. And plantain, it's an invasive exotic, but it's also really important for Taylor's checker spot because it's way more common than the other stuff, and the other stuff sometimes is almost absent from sites that are really important in Taylor's checker spot populations. So, Plantago. Harsh paintbrush is the most important native host. It's not terribly common anymore where there are also Taylor's checker spot populations, although it's increasing because we plant it a lot. And then golden paintbrush was very, very uncommon, but it's had a huge upswing in restored sites where we're seeding it hundreds of thousands of plants now. We don't really know how good of a host it is for Taylor's checker spot, but it's now at a lot of the same sites where they're also reintroducing Taylor's checker spot. And there's this interesting issue where these 2 species hybridize with each other and you get this really cool orange paintbrush. It looks beautiful, but it's a problem for recovering rare species because if you're trying to recover golden paintbrush and then it interbreeds with harsh paintbrush and you get all this orange stuff, you're not really sure if that counts. So they got to keep them separate, which means there's a site that has just golden paintbrush. Is that okay to put Taylor's checker spot there too and try to restore Taylor's checker spot, or do we have to keep it on the harsh paintbrush? So we don't really know which of these plants is better or worse than the others as a host. It's a little more complicated than that, but that's the beginning. So again, harsh paintbrush, that's the red one. Golden paintbrush, the yellow, golden one, and plantain. Okay, so it's been really interesting. One of the really interesting things about checker spots and their host plants that we've learned, especially in California, is that sometimes their host plants dry out as the dry season, when the rain stops in April or May or whenever. The host plants dry out, March this year. The host plants dry out. So here's the harsh paintbrush on May 15, and here it is in the middle, like a month later. And it goes from this lush food plant to this withered stick, right? And in a way, at least in some populations, and definitely in California, there's this race against time where pre-diapause larvae are trying to eat their host plants and trying to eat them before they dry out. And if a host plant dries out before the caterpillar gets to like 4th instar, and it doesn't have anything else to eat, it'll just die. And that's actually in California populations, like 90% of them can just die because their host plants all dry out. So we don't, so Stu Weiss did a lot of work with this, and Mike Singer and a bunch of other folks. Stu's been around the prairies a couple of times, so he's fun to talk to. So we don't really know if that's an important thing, if that host plant senescence is important in Washington populations or if it's no big deal. But the plants definitely dry out around the time that the larvae are still feeding, so we don't know if that's important. So my goal was to compare these 3 host species and figure out, hey, do they all senesce at the same time, or are they different from each other? Do they differ in terms of their iridoid glycosides that they produce and concentrations? Do the larvae do the same on all 3 of them, or do they survive better or grow bigger on one versus the other? And then if we look at like an environmental gradient from really dry, harsh spots to like a nice, moist mesic site, are there differences there where like maybe the larvae survive better in one than the other, or the plants dry out faster or slower in one than the other? So we wanted to figure all that stuff out. So what we did, and a lot of this work is myself and Peter Dunwoody, who you all might know, and Mary Linders, who is with the Department of Fish and Wildlife, and John Bacher, my advisor. We set up these plots, and this is an area of glacial heritage preserve down by Olympia, south of Olympia, and West Rocky, and Tenalquah Prairie, which is a little further, it's kind of by Lacey, near Joint Base, Lewis McCord. And all those red dots are plots that had harsh paintbrush, the golden ones are golden paintbrush, and the green ones are plantain. And there's 15 at each of these sites, and then if you add them all together, there's 15 for each of those species. And they capture a range of environments from like really dry south sides of slopes to like a nice, moist, shady environment. Each plot looks kind of like this. It's got 5 plants on it. And we built these little playpens around them so that the caterpillars could run around in them, but it would be hard for them to get out. And we could cover them up if we had a lot of birds eating caterpillars. And then basically what we did is we took a little egg cluster and put one in every one of these plots. So we got the egg clusters from the Oregon Zoo and the Women's Correctional Facility, and they gave me these little tubs with drops of eggs. And then we went and put them on all these plants. So we kind of attached them with paper clips because they haven't been laid naturally, and then they would hatch and eat the plant. So does that make sense so far? Pretty good. Good. Okay, and then we took all kinds of measurements. So for the plants, we wanted to know how quickly are they drying out? So every week, how brown are they basically? How big are they? And then we took leaf samples so we could know their nutritional and chemical information. And then we measured the soil moisture every week, and I put these little data loggers in all the plots that measure temperature every hour or so. And then the slope and the aspect and the soil texture is really sandy, is it full of organic matter, etc. The nutrients and all kinds of stuff. So lots of environmental information. And then for the caterpillars, every couple of days we tracked their survival, what instar they were in, how big they grew, and what their, eventually we took a subsample to be ground up and have their iridoid glycoside concentrations measured. Because that's really important. It's unfortunate when we have to squish a bunch of caterpillars, but it also tells us whether they're defended against predators or not, and whether that differs depending on what species they're eating. So really important.
Except human predators.
Yeah. I ground them up anyway. To be fair, these were all, first they meet their recovery targets, and then they get a few more eggs beyond what's actually needed and what they can put out logistically, and they give those to me. So these are, and you'll find out I don't kill nearly as many as nature kills. So here's what they look like out on their plants. And they really make messes in the castellia, which is fun to watch. And when they get towards fourth instar, they're a little bigger and more photogenic. And here's what we found. Only 6% of those eggs made it to fourth instar. We had 45 plots. We had 0 survival at 29 of them, where just everything died. And then a few where they survived. And in a sense, that's okay, right? Because a female lays thousands of eggs, so we're not expecting like 90% survival. If we did that from one year to the next, we'd have like 1,000 times more butterflies, which would be kind of awesome, but maybe too many. And there was a little bit of variation in survival depending on which host they use, but it's probably not statistically important. And this is sort of the take-home figure right now. This top panel here is how much live tissue is left on the plants. So this is the day of the year on the x-axis. So this is basically April, and this is June 29 right there. There's June 19, June 9. And you can see the plants are totally green and red and not brown in the beginning, and then they go and they dry out through time. And you can see harsh paintbrush and golden paintbrush dry out pretty significantly. Golden paintbrush might dry out a little more quickly. This is CALD, Castellia secta. Sorry about the code down. So you can see they turn really brown. Castellia secta might turn a little browner. And plantain pretty much stays green way into the summer. So senescence is not an issue for plantain. It might be an issue for these other two. But here's what's really interesting. This is a curve of the larval survival. So at the beginning, 100% of them are alive. And at the end, less than 10% of them are alive. But what's really interesting is almost all of them that died when they were eggs or first instar or maybe second instar. And when they were dying in droves, plants are actually still green. So that California thing that happens where the plants all dry out and then everybody starves, at least this summer, that didn't happen in Washington. So now we know. We need to measure that multiple years. But this was a really dry year, and that didn't happen. Larvae found all kinds of other ways to die that didn't have to do with which host they were eating, and didn't have to do with how quickly they dried out.
Because they died just as much on the plantain.
Pretty much.
Like,
Yeah, and now we're still kind of analyzing. There's this huge, massive data set that I'm trying to work on. But what we've concluded so far is the bottleneck in survival happens a lot earlier than we thought maybe it did. And we think it has to do with way more with predators than it does with the quality of the plants that they're eating. So we're trying to figure out how to redo this experiment next year and be really careful about doing predator exclosures and sampling the predator community and trying to catch them in the act. Because as much time as I spent staring at those plants and at those caterpillars, I never saw them getting eaten. It could have been happening at night or, late in the evening after I went back to Seattle or something like that. So we're trying to figure out what to do next. But what I think happened is there were thatch ants. There are thatch ants everywhere out there. Those are the ants that make those really big mounds, And they eat lepidopteran larvae, and they were all over in some of these plots. So I've been mapping the thatch ant mounds and trying to figure out if they have paths that go near some of the.
You didn't see any. You didn't see them eating.
No, we would just come back out and like the little bit of leaf that had the eggs on it, the eggs would all be scraped off, and the little flake of leaf would just be sitting there with no eggs on it. So something came by and just like gobbled them all up. So that's kind of too bad because we framed this huge experiment in terms of all about host plant quality. But what we found out is really important, right? If we want to conserve Taylor's checker spot, well, golly, let's think about predators a little bit more. So that's been really interesting, and we're just now going to be meeting with a bunch of partners to figure out what to change about this experiment so that we can measure the predators, and then also exclude some predators so that we have a whole bunch of larvae that make it to later instars, and then we can measure what happens with the host plants too. So we're trying to figure that out right now. So that's just a little snapshot into that experiment. Yeah.
So, obviously, since you're planning first instars, they all, made it from their egg to their first instar, but is there any kind of...
Oh, I actually planted eggs.
Oh, you planted
Yeah.
eggs? So, do you know what percentage of your eggs didn't hatch? And what about, like congenital abnormalities? Is there such a thing with instars and butterflies? Do you know anything about that? That would prevent
That could happen, although these were being raised, these individuals had other stones being raised in captivity where I would have heard
So they were
they all died.
Yeah.
They do fine in captivity. So it was some sort of species interaction. Another thing that could happen to them is they just dry up because it's a really hot spring. But then you find their little withered corpse. And we never did. We'd see a little web full of caterpillars one day and come back 3 days later and it's all cleaned out, just gone. So probably predators. We saw, spiders and mites and parasitoid wasps and all kinds of things that it could have been. So, yeah.
Is that a native ant?
Yes. Yeah. I've been trying to figure. I didn't know much about them. I've been following them around on the prairies these last couple of weeks, and a lot of their mounds, they have a path that goes to a Doug fir tree, and they all march on that path and they go straight up the tree, and they're tending aphids up there, collecting honeydew from them. And, so that's what they seem to do most of the time. But I know they also eat a lot of caterpillars and random insect eggs. So I'm really curious about them right now. I'm just learning.
How big are they? Are they like sugar
They're
eggs,
pretty big,
or
like
bigger than-
a little smaller than a centimeter long. They have a little red on them. Yeah. Great. Well, thanks for listening, everybody.